Ensuring a stable food supply. It's just fundamental, isn't it?
Yeah.
For our economy, for society. And, well, there's some really interesting research emerging from Germany now.
Oh, yeah. What's the focus?
It's tackling plant viruses. Specifically, they've developed a more effective type of double-stranded RNA.
Ah, dsRNA for
Right.
dealing with viruses like CMV?
Exactly, Cucumber Mosaic Virus.
Hmm.
And they're seeing a huge reduction in the virus, like up to 80%, which is way better than what we have now.
Wow, 80% is significant. So what's the core idea? How does it work?
Well, the really cool part is the precision. It uses the plant's own defense system, RNA silencing, but in a much more targeted way.
Right, RNA silencing, a natural defense. And this German research is refining that for CMV, you mean?
Precisely. And CMV, it sounds specific, but gosh, it's everywhere.
It really is a truly global issue. It hits something like, what, over 1,200 plant species?
Yeah. Things we eat every day, cucumbers, melons, bananas, pumpkins.
Yeah.
The list just goes on.
And the symptoms are pretty clear usually. Yellowing, those weird mosaic patterns on leaves, stunted growth.
You see it, and you know something's wrong. And it spreads so easily.
Mm-hmm. That's the key challenge. Mainly through insects, right? Aphids mostly.
Yeah, these tiny sap-sucking insects. Apparently, around 90 different aphid species can carry it. They just feed on one plant, pick it up, move to the next.
Makes it incredibly difficult to control once it takes hold.
And the impact, particularly somewhere like India, is just huge. You hear figures like 25–30% yield loss in bananas.
And even higher in things like pumpkins and cucumbers, right? Up to 70% infection rates sometimes.
Exactly, and you end up with discolored, stunted plants, deformed fruit, and there's no real cure for it currently.
It's a massive problem. If you look globally, the FAO stats are quite sobering. Almost 40% of crops lost each year to pests and diseases.
That's staggering. What does that translate to financially?
Over $220 billion annually. And plant viruses like CMV are responsible for over $30 billion of that. So this German research, it's potentially a big deal.
Okay, so let's unpack this RNA silencing a bit. It's something plants do naturally.
Yes, it's an inherent defense mechanism. When a virus infects a plant, it often produces double-stranded RNA, which is kind of a red flag for the plant.
The plant sees it as foreign.
Pretty much. It triggers these special enzymes, dicer-like enzymes, or DCLs. Think of them as molecular scissors.
Scissors that chop up the viral RNA.
Exactly. They slice that dsRNA into tiny fragments. These are called small interfering RNAs, or siRNAs.
Okay, so you get these little siRNA pieces. What then?
Those siRNAs then basically act as guides. They load onto a protein complex and guide it to find matching sequences in the virus's own RNA.
And destroy?
And destroy it. It effectively silences the virus, stops it from replicating.
Hang on. If plants can do this naturally, why is CMV still such a menace?
Well, that's the catch. The natural system isn't perfect. For one, not all the siRNAs the plant produces are actually effective against the virus.
Some work better than others.
Right. And also, viruses like CMV mutate incredibly fast. They can change their genetic sequence just enough to evade those siRNAs. The plant's defense can't keep up sometimes.
Okay, so nature needs a bit of a helping hand. That leads us to human-developed tech, like host-induced gene silencing. HIGS.
Yes, HIGS is one approach. That involves genetically modifying the plant itself to produce specific dsRNA that targets the virus.
But GM tech has its own hurdles, doesn't it? Regulations, costs.
Exactly. Public acceptance, regulatory frameworks can be complex, it can be expensive, and there's always the chance the virus could develop resistance to the engineered trait, too.
So what's the alternative? I think you mentioned SIGS earlier.
Right, spray-induced gene silencing, SIGS. This is different. Instead of modifying the plant, you just spray RNA molecules onto the plant's surface.
Ah, so you trigger the plant's natural silencing pathway from the outside without any genetic modification.
Precisely. It has huge potential advantages, potentially cheaper, easier to deploy, more eco-friendly perhaps.
Sounds promising. But it must have limitations, too.
Well, yes. The challenge with traditional SIGS approaches has been, kind of like the natural system, that the dsRNA you spray gets processed into a whole mix of siRNAs, many of which aren't very potent against the target virus. It's a bit scattergun.
Okay, so inefficiency was the problem.
Largely, yes. And this is where the breakthrough from that German research comes in. They figured out how to create or enrich the dsRNA they spray, so it predominantly produces highly effective siRNAs against CMV.
So they're not just spraying generic dsRNA, they're spraying effective dsRNA that's preloaded, in a sense, to generate the most powerful silencing response.
That's a good way to put it. They're making the SIGS approach much more targeted and potent, overcoming that previous limitation, giving the plant the best possible tools.
That really does sound like a significant step forward. More efficient, no GM.
It could be a game changer for managing viruses like CMV, offering a more reliable, perhaps more acceptable tool for farmers.
So tying this back for our listeners, especially you UPSC aspirants tuning into this deep dive, this work, highlighted in the Hindu, is right at the intersection of biotechnology, agriculture, and food security.
Absolutely. It demonstrates cutting-edge innovation tackling a major global challenge with potentially huge economic and environmental implications, particularly for countries like India that are heavily impacted.
It definitely makes you think about the future, doesn't it? How technologies like this could reduce our reliance on traditional pesticides and bolster food security globally. A provocative thought there.
Indeed. The potential ripple effects are quite broad.
Well, we hope you found this discussion insightful. If you did, and you want more deep dives into topics relevant for your UPSC preparation, please do like this, subscribe to our channel, and definitely leave any questions or thoughts in the comments below.
Yes, your feedback helps us choose future topics. Thanks for joining us.